Hello everyone, today we will talk about the characterization and annotation of novel FM cluster bacteriophage Ottawa. So bacteriophages go through two main cycles, lytic or lysogenic. In lytic state, phages can infect and lyse the bacteria. In lysogenic cycle, phage DNA gets incorporated into bacterial genome, resulting in bacteria replicating with phages DNA. Lysogenic phages can become lytic at later stages. There are also three main types of bacteriophages based on their appearance: Cephaloviridea, Myoviridae, and Podoviridae.
Our research is part of the CPH program, and essentially what we do is that we collect soil samples from local environment and have an opportunity to isolate our very own bacteriophage. We first began by collecting and purifying our soil sample. We promoted the growth of host bacteria, Orthobacter globifermis, by adding it to a petri dish that contained pica media. The reason why we promote the growth of bacteria is because it allows the phage, which is host dependent, to proliferate. So we increase the number of bacteria, increase the number of phages, which means we have a higher likelihood of extracting something. After a series of tenfold dilutions, we created wet plates, allowing us to calculate the concentration of phage that we have in our sample, and eventually reach our goal in reaching a high titer lysate that would allow us to perform all sorts of experiments, such as and not limited to restriction digest, DNA extraction, and imaging our phage. We then had the chance to annotate the genes in Ottawa through using a combination of databases such as HHPRED and CVI BLAST-P and a star rater report. We were able to determine the relative gene functions of the assigned genes. We then determined the synteny between the other phages that had similar gene functions. In the case for most of our genes, we determined the synteny between the phages Ottawa and Karsho. Few genes, such as gene 3 that encodes for a tail terminator function in Karsho, while in Ottawa encodes for a minor Caspar protein, were different. But for the most part, the relative gene position and functions of the genes in Ottawa and Karsho were similar.
Here's the characterization. So we'll discuss location, plaque, and phage family. So the sample containing Ottawa was collected on the Ottawa riverbank. Ottawa's plaques are considered small and cloudy, which suggests it undergoes lytic and lysogenic cycles. And upon time imaging of Ottawa, it was decided it belongs to the Cephaloviridea family in accordance to its 270 nanometer tail length with infective specificity against Orthobacter globifermis. Upon digestion by restriction enzyme, only 2 of 8 acid enzymes were able to partially cleave Ottawa's genome, which were HEY3 and BSTU1, similar to the behavior of the Karsho genome. This may indicate the presence of 2 amino adenine or DNA methylation modifications. This is hypothesized in Ottawa, but future experiments with restriction enzymes will allow us to make definite conclusions. Additionally, these properties may confer major advantages to phages in protecting against bacterial nucleases.
So Ottawa is a Cephaloviridea phage with a long non-contractile tail. As such, it contains something called the tail assembly chaperone, so abbreviated as the TAC. So the TAC is an essential protein that is found in most Cephaloviridea phages, which chaperones the tape measure protein for the assembly of a tail. TAC genes are often found in between the genes encoding the tail tube and the tape measure, and there are 2 forms of the TAC that exist. So we have a smaller form and a longer form. To see both these forms, we will sometimes get a programmed translational frame shift that we can see in the genome. And we know that there is a frame shift somewhere which can be found by finding the slippery sequence. And in our case, that is the GGGAAAAA. Essentially, the ribosome slips from the GGA to the GGG. So this is a -1 frame shift in this case. However, we suspect that there isn't a frame shift. Instead, we believe that the gene 43, so which is a long fragment here, is being called a TAC because the majority of the sequence includes gene 42, which is actually the TAC. So we thought it would be beneficial to do wet lab experiments to actually confirm if this frame shift is actually there. So what we did in the lab was that the gene 42-43 fusion TAC gene, so that was first initially amplified and inserted into a PGEX6P1 vector. And so this vector is then transformed into the DH5 alpha E. coli strain, and it grew overnight. We would then transform the plasmid to Rosetta E. coli to express the protein better, and then we'd purify the TAC protein and then use mass spectrometry to confirm whether or not there is a frame shift. So for future directions, we want to create a lysogen for Ottawa, where the bacteriophage stably integrates into the chromosome of the host bacteria, Arthobacter globiformis. We want to test Ottawa's ability to infect bacterial species other than Arthobacter globiformis. And we also want to do a restriction enzyme digest to determine if the FM cluster of phages lack restriction sites, which results in the generation of protection against bacterial nucleases. Thank you for listening.